So let me start first by highlighting some of the things that probably many of us already know. For example, cancer, which is one of the leading causes of morbidity and mortality worldwide, is much more prevalent in areas of the Western world that has a growing and older population. And so if you think about that trend of the population becoming older and having additional comorbidities, it is clear that to address this effectively, the same standard measures that are working for everyone are not going to work for any particular individual in a very precise and effective way. And so it's very important for us to recognize this trend and to understand what are some of the ways that we could effectively address this growing problem that is going to require much more investment in new technologies to address these pressing needs. For example, when we think about cancer, we need to understand what are the drivers of cancer progression, what are the risks associated with developing cancer, and therefore develop screening and early diagnostic tools to develop develop an understanding of what are the markers of cancer risk and progression. And so in that approach, what we're really seeking to understand is how can we detect these molecular signatures that are very specific and unique to each individual, and be able to recognize that small piece of data, that signal, out of noisy, huge data sets that are generated every day for millions of patients around the world. And what that has to do with are new technologies that allow us to look directly into the DNA, understand the genes and RNA transcription, and look at the sequences of individual cells and tissues as this disease develops over time. And so what we're looking at is really a collection of complex data sets that include patient-derived cell lines, patient-derived xenograft models or animal models with human tumors in them. So the cell lines and the patient-derived xenograft models are some examples of how we can study the basic biology behind this disease, but also we can incorporate the clinical trials and some of the big data collected from real patients where we can find some signature information and start to ask questions. And eventually what we're seeing as a result of that is more precise and targeted therapy for many diseases, including cancer therapy. And a lot of these are actually changing our ability to treat people more effectively based on these molecular signatures, combining our understanding of drug metabolism with our understanding with genetic predisposition and with an appreciation of disease progression or the basic biology behind how this disease goes from non-malignant to more and more malignant and invasive, and eventually metastatic cancers that ultimately lead to death. And so this appreciation of data really is central to our ability to be able to provide more effective and precise treatment of patients. Now, unfortunately, what that brings us to are some big challenges with data that is very diverse, very different because it was generated by thousands of labs around the world, different hands, different methods and approaches. It was generated from many different types of people. We don't know all the information about those people. So really, it's a problem of big data. And this problem of big data is a different type of a challenge than what clinicians and biologists are used to. So biologists coming from a life science background are used to clean data, experimental data, data that had a design, data that had very specific parameters included in how the data was generated, and therefore you can test your hypothesis by looking at the signal in that data. And for clinicians, I think it's even much more challenging than that, because what they're used to be dealing with is data that is not high throughput. It is data that is obtained from lab reports. Those lab reports come with a medical history, with a medical background, and are contextualized by medical knowledge that individual or that location really has. And so as opposed to that, when we come to this challenge of big data in the clinical application, we need to appreciate some things that are coming really from the research side, as well as understand the context for many different patients that we could potentially not ever interact with, but they're really repositories of raw data that we can mine and extract some meaningful signatures from. And potentially that could help us answer some important questions about the patients that we interact with. And so at the baseline of this, I think, is one of these technologies, and this technology is called next generation sequencing. And I think that for many of you, probably you've heard a lot about genomic data. You've probably heard about the applications of genomic medicine in a variety of different diseases. But I think what is important is for everyone to have that skill set and have that expertise in being able to look at the data, understand its quality, be able to find specific signal, and then to also lead to biological or even clinical interpretation of that signal. And that's really what this program is going to be about. So we're going to introduce you to a number of different ways to study this kind of data, analyze sample projects that we've prepared and curated for you to understand the specific questions and the applications of that data set, and then introduce you to some of the methodologies that will be useful for you to practically work with such data sets, understanding genomic variants, understanding gene expression and the variation in gene expression based on differential gene expression, looking at the annotation and interpretation of how these differentially expressed genes could be combined into specific biological pathways, and how can we really understand the biological pathways in the context of a particular disease or a biomedical question. And so one of the repositories that we will turn to is what's called the Cancer Genome Atlas, or TCGA. Now, TCGA is a very important repository of thousands of samples collected from patients that are that have many different types of cancer, so coming from different tissue and body sites. And what it provides us is some kind of an appreciation for multi-omics data and its role in understanding the disease stratification. So how is disease variable between different patients, patient stratification, how different patients are represented in this data set. They come from different ethnic backgrounds, different age groups, different sexes, genders, and how can we use this data to understand the disease in a much more precise way. So this is a really unique repository that actually anyone can gain access to and start downloading that data to be able to ask research questions, finding associations between treatment, between risks, between different types of omics data. And so what we will address in this program is how to integrate such data to be able to find patterns that are relevant to different types of cancers. We'll also take a look at some of the algorithmic approaches that allow us to apply machine learning to extract signal from data sets that were generated in a high throughput way. And so one unique aspect of high throughput experiments is that the data has been generated in an automated way, and we can generate such data from cell lines based on, for example, response to treatment. So ultimately, the goal there is not to understand the molecular mechanisms behind efficacy of drug treatment, for example, but it's really to find associations to understand some signal, some kind of biomarker of efficacy. And that kind of information can then help us assign better treatments to patients, not by understanding and studying each patient individually, but actually running some algorithms that could bring out that information in front of the person who is delivering the treatment and guiding them in their decision making. So there we will take a look at how actually both of these are integrated. So we'll take a look at animal model experiments, cell line experiments, and then compare them to the variability in patients, looking at research articles that describe the data sets and trying to extract signal that is aligned between cell lines, animal models, and ultimately addresses the challenges with individual patients. Now, in addition to cancer, many other diseases are going to be on everyone's mind. Obviously, we live in an era of a pandemic, and so it's important to appreciate the role that viruses play, not only in the pandemic that we're experiencing right now, but also in many other conditions. So a lot of times we think of infectious diseases like SARS-CoV-2 as a virus that is causing a pandemic. But surprisingly, many different viruses are involved in a variety of diseases. For example, Epstein-Barr virus is involved in many cancers. Hepatitis can lead to liver cancer. So there's many diseases, including neurological diseases, that could be associated with either the direct effect of the infection or inflammation that is caused by chronic infection. And so we'll take a look at how other types of diseases could also be studied using technologies like next-generation sequencing to understand the disease itself, the immunology or the host response to that disease, and the variability in disease manifestation in many different types of patients because of what is actually going on in different tissues and cells that we look at through the lens of data. And so that's why I think that bioinformatics is really central to our ability to improve medicine and improve treatment that is more effective, less toxic, and guided by our understanding of these molecular signatures. And so to develop bioinformatics skills, you need to have some sense of the biology, computer science, and the statistical methods that are used to analyze such data. And so we hope that as a result of this program, what you will be able to gain is an understanding of a variety of bioinformatics algorithms and software, to appreciate the different databases of data that includes genomic data from cancer studies, infectious disease studies, and other diseases. You will appreciate the analytical techniques and approaches that are needed to analyze data, and you will be exposed to a variety of data science skills that, if you are interested in, you can pursue further in other types of training to develop a practical skill set that is needed for the age of big data. And so as an example, we have here a few omics data in infectious disease research that I think we'll also cover in many of the sessions that we'll discuss, that will include an overview of viral genomics and evolution, immune response biomarkers, as well as an overview of microbiome and its role in infection. Now, obviously, a lot of what I'm saying seems like too much to cover in a single program. And so that's why what our team has done is collected a number of program resources that we're going to be making available to all of the program participants to make this a more streamlined and effective use of your valuable time. So we will not only cover these live sessions like we're doing today, we're also going to provide you access to our training materials, fast big data analysis tools that you can run via a visual graphic user interface, curated project data sets, so you don't have to spend all of your time finding data, but you will be given some data sets that you can quickly go through to understand the point and the connection between the analysis and the biological relevance, as well as these interactive sessions that will be guided by mentors that I will introduce in just a couple more minutes. But before I do that, I want to introduce you. I know that some of you are probably already familiar with this, but I'm going to briefly speak about our big data analysis platform that allows anyone to quickly go through some of the important methods in bioinformatics and try them out on different types of projects. So we will, in a second, go into how to register and how to create an account. And once you do, we will take a look at this big data analysis platform so that you can understand that you can both learn and then use this platform for actual research projects that you might have. And so here you will find a variety of different types of omics data that you can analyze. You can run demo pipelines to understand the logic behind some of the different analyses that we will use. You will also see how you can share your analysis with someone else so that someone like a mentor can provide you with technical information about what went wrong in your analysis. And it also includes user-friendly tools to apply data mining and annotation of biological information so that you can get quick insights about the data sets you're analyzing. So to really see how all of this works, let's go to learn.omicslogic.com and let's start by creating an account. And that will allow us to take a look at some of the data sets and some of the example or projects that we will cover in the program. Okay, so to go there, so Narika, if you could please share the link. Okay, so when you get to this program, when you get to this portal, learn.omicslogic.com, you can create a free login. This login is going to be free for anyone. You don't have to participate in the program. And in fact, if you would like to use this asynchronously, you can. Okay, when I share my screen, let me stop sharing just real quick. So when you create your account, you will gain access to both the coursework and the platform. Okay, so here you can see that you can create an account for free. Those of you that don't have an account, you can do that now. I'm just going to sign in. And once you're in, I will show you what are some of the resources that we will be using. And later on, I will pass it on to Sonanika to also look at how to register for the program itself. Okay, so please let me know once you're in. And once you have an account, you can put yes or number 1 or something like that in the chat box so that we know to proceed. Okay, so I see people signing up right now. So if you have not signed up yet, please sign up. I want to demonstrate how the program resources are going to work. Okay, thank you, David. All right, thank you, Dr. Bashir. Okay, so hopefully you've created your account now. You will find that your account comes with a personal profile. And in this profile, the first thing that you want to do is you want to finalize your name, your profile picture, give us some description about you. And this is going to become your personalized center of your learning experience. So here you will find the courses that you are taking, your progress. If you've completed some course, you will see a certificate. You will also see your certificate separately here if you would like to download them. But importantly, you're also going to have these programs here. And so once you join one of the programs, again, you will see what are the specific things that are associated with each one of our programs. So if you go to the Programs tab, here you will find all of the upcoming programs that we have. And in this particular programs on precision medicine, we have included a number of courses and projects that are going to be relevant for your ability to learn and apply bioinformatics techniques to a variety of questions and examples. So here you will find projects like the Cancer Genome Atlas, an example with liver cancer project. You will see some projects on COVID-19. You will see some technical skills that are going to be useful as you learn to integrate, visualize, and analyze your data. And you will see a number of explanations about some important machine learning techniques that we will cover in the course as well. So take a look at this, and later on, Sonanika will explain exactly how this program is organized to continue and pass it on the main component, I think, that differentiates this program from some of the other programs that you will find online on similar topics is that this is not just access to these online resources. This is access to training material, big data processing, which you will typically not find in anywhere else. And this is going to give you some real examples of methods to process your data. And so to briefly introduce this resource, all of you that have an account, you can use the same account to access a demo pipeline that I will briefly demonstrate to show you how easy you can go from learning about a specific method of analysis to actually using that method of analysis. And so to do that, I will go under the transcriptomics section, and I will open a number of demo projects that we will use during this program. So one of them is called the Cell Line Project. And so this is a project where they took a variety of breast cancer cell lines and performed gene expression analysis. So they generated transcriptomic data to understand what differentiates between different types of breast cancers. So here we can quickly process the same data, and as we go through the analysis, appreciate what are some of the logical steps that you need to take to go from raw next generation sequencing data, FASTQ files, all the way to getting a table of expression. So the main steps are pre-processing, mapping, and then quantification of expression levels. And what I'll do today is I'll build a very simple pipeline that in reality, if you were to try and make this pipeline on your own, might take you maybe a few hours, a few days, or even a few weeks, depending on your level of expertise. But you'll see how quick and simple this could be right here. So first of all, what we'll do is we'll clean up those raw reads, and we'll make sure that we remove the technical adapters from library preparation. We will then address the issue of PCR amplification that could bring in some technical variation that we don't want to confuse with biological signal. Then we will align these reads to the reference genome using bow type 2. And then we will quantify the levels of expression to get a table of FPKM gene expression using RSEM. Once we've completed this pipeline, I can click on End, and I will take a look at some of the output data. So it's a real quick process to go from having that raw data to then analyzing that data, and you saw some of the variations to the pipeline that you can make as you go through, and then getting a result so that you can appreciate the inputs and the outputs very quickly. Once you get those results, importantly, we have all kinds of additional methods to mine this data, visualize this data, and even integrate multi-omics data to quickly understand the relationship between gene expression, genomic variants, maybe microRNA and long non-coding RNA, as well as some clinical information.